So welcome, and I'm going to talk for about 30 minutes on some of the work we've been doing at UCL in our plastics hub, which we've created over the last few years to look at how we might start to recycle plastics using synthetic biology, using enzymes and microbes. I'm John Ward, and I'm, as you see there, Professor of Synthetic Biology for Bioprocessing. Bit of a long title, but some of the things like synthetic biology I'll talk about near the end of the talk. So I'm going to talk about plastics, and you probably know plastics are polymers. Plastics is a sort of more layperson's name for polymers, and over the last 40 to 60 years, polymers have replaced many of the natural materials we used to use. And natural materials that commonly use, things like glass for drinks bottles, and that's been replaced by this polymer called PET that you've probably all got it maybe in your hands now, these polyethylene terephthalate polymers, which have brilliant properties in being able to be strong, light, and they can actually retain gases such as fizzy water. So they retain the gas in. So they actually have a lot of very nice properties. Polythene, you've all used polythene bags at supermarkets, and those have completely replaced cloth or natural materials like wicker baskets that we used to use in, well, I remember the old days. And of course in textiles, we're all wearing or do probably wear textiles that are natural, but also polymers like nylon, lycra, which gives things a stretch, very interesting polymer, and polyesters, which we'll talk about quite a bit in this lecture. And these have replaced cotton and wool for clothing in many aspects, and they have many good properties. There are other polymers that are in everyday use, like polymethyl methacrylate, that's Perspex, and that's replaced glass in many applications in small screens and viewing chambers and things like that, because it's tough and is transparent. And then we've got loads of other polymers, polystyrene, polypropylene, and many others, and these have replaced a lot of things like paper, aluminium containers for food use, and structural materials now in things like cars and in most of the electronics and things you see around you all the time. Huge numbers of polymers in those. It's because they have a lot of advantages. There's a huge saving in weight, for example, between PET and glass. And that saves energy and transport costs. And that is really very significant if we're thinking about not using PET anymore and returning to glass. The energy that's used to transport glass bottles is enormous. So we have to really consider that in these days of really trying to get carbon usage and CO2 usage down through fuel. Many different aspects of life now rely on this very great saving in weight of polymers.
Sorry to interrupt, John. I just wonder if there's a piece of paper next to your microphone. There's a bit of interference coming through.
Okay, am I rustling?
Yeah, there's a little bit coming through.
Okay. Polymers also have a big resistance to degradation, and we're going to look at that in one or two slides here. That's a great advantage when we're talking about materials for housing, structures, things that we don't want them to decay away in 5, 10, 15 years. However, that in itself is a problem, that resistance to degradation. And they're great versatility. Polymers have huge numbers of advantages compared to natural materials because there's many more of them. So they have different properties. They can be shaped and molded or in a factory. They can take different dyes more easily. So there's a lot of advantages. Now, some of those advantages, I talked about this resistance to degradation, is actually also a disadvantage when it comes to things like how do we dispose of them at the end of their life. And there isn't really, or there was never thought to be, are considered a real end-of-life strategy for plastics when they were first being developed. And because of that, they build up in the environment. So in the early years, that was all okay because people just didn't think about that. They put it into landfill. But in Europe and certainly the UK over the last 5 to 10 years, there's no space left. There's very limited space for landfill. We don't recycle as much in the UK as they do in some countries in Europe, but we are doing that now. But even so, a lot of plastics ends up in places like the oceans and watercourses, and that's where it does its harm. And I'm sure you've seen those pictures of plastics in the ocean and animals in the ocean eating and being harmed by plastics. And of course, any plastic that's burnt in just general fires or in sort of waste to energy conversion leads to a net CO2 increase because plastics are based on oil. And right at the end of the lecture, I'll talk about maybe a future where plastics might be still being used, but they're based on different inputs. And here's just a little slide that actually quantitates some of the plastics found in European fresh waters, and it's from the Earthwatch Institute. And there are some numbers there, you can add them together. So plastic bottles, plastic bags, food wrappers, food containers, and that's well over 30 to 40% of plastics being found in fresh waters. So let's have a think about this non-degradability. This is a picture I took last year. We went down to an old landfill waste site on the banks of the River Thames, further out towards the sea than you might be familiar with on the Thames. So that's where it gets very wide and flat. And in the 1960s, there was a big domestic and probably a commercial landfill site, big hole in the ground, probably an old gravel pit, and it was filled with refuse from London. And then it was covered over, and from the 1960s onwards, so we're talking about, what's that, 60 years now, it's not been used. It's been covered with soil, and then that's gradually built up. Now, the storms and the high tides have eroded the riverbank along this part of the Thames. And so what was once covered is now being revealed. And you can see this lovely, almost archaeological layer of plastic and other rubbish. A lot of glass there that's all fallen onto the seashore. But the plastic is largely as it was when it was put in there. It's become a bit crushed, but it's not decayed away. Very visible. So that's from the 1960s, 60 years old, nothing's really degraded at all. And that's even in the days when much less plastic was used. So there was no PET water bottles then, it was all glass. There's lots of glass in this waste site. No lycra, no plastic food containers, but there was polyethylene that was well in use then for building materials and other things, polyesters and PVC, very commonly used in those days. But the amount of plastic used now is much more. And this is a site we carried out some microbial sampling in October. So his next picture is where we went down to the Thames. We knew this landfill site had now started to be revealed and eroded away at the beach level. And we wanted to try and sample for some new bacteria that might be able to grow on some of this plastic, albeit very slowly. So this is a picture of Maria Bourne, Dr. Maria Bourne, who's one of my postdoctoral researchers in my group, sampling some soil and some material from in and around these plastics that are under the soil here. And that's just a picture at the bottom of the rather flat and open, bleak expanse of the Thames, near the Thames estuary where we were. So I'm going to talk about this polymer that we use a lot of and is very obvious and very evident because all our plastic water bottles are made out of this, but many other containers as well. It's found in a lot of food containers you buy in the supermarkets. So it's widely used. We call it PET. It's got a longer name. We'll look at that in detail in a few minutes. Polyethylene terephthalate. Bit of a mouthful to say, but we call it PET. And it's used in textiles. So polyesters are often this polymer. So not only in food use, in water bottles, but it's used in textiles as well. And there's huge amounts of it entering into the system every year. So it's estimated there are 3.3 million tons of PET bottles alone. That's not including the textiles and other uses of it. And that's in the European market in 2017. So you can see it's a huge number just in that market for the bottles. If you extend that to the whole world and other uses, it's very, very large. And then it turns out you can find PET enriched in nature. So this is a way of just saying it forms plastic waste. And we have it all the way from the whole bottles to plastic fragments. And because it's been around for a while, then it's degraded and abraded into little plastic fragments called microplastics. And these are found in all the oceans now and terrestrial environments. Now, it turns out that the PET bottles are very easy to collect in waste and domestic waste, and the food containers as well. And they are 100% recyclable if they're collected in these big waste collection facilities. But it's by mechanical recycling. So that's quite distinct from what we perhaps want to do in the future. So I'll just talk about mechanical recycling of PET. So you can see the diagram here. We've got feedstock, that's oil, crude oil, and it's refined and made into the various monomers, and then factories will take those monomers and build them up into the plastics, into the various products. Humans use them, and then they get taken into the waste streams, collected into these big waste recycling facilities. It's shredded, and we get mechanical recycling. And that's basically just sort of breaking it down into bits, fragments, and then they can be used again if they're kept into their specific polymer waste streams. So for example, if you collect lots of PET bottles and food containers, they can be kept separate from the other polymers and waste. They can be collected, sent to a special factory that then deals with mechanical recycling of PET. It's shredded and melted and made back into PET bottles. So we've got some kind of cycle going on there. That's good. But there is a gradual loss of the properties of PET if you keep doing that. So you've got a loss of the clarity and strength and length of the polymer. So in any facility that's doing that and making new PET bottles, you always have to add some fresh PET polymer from the petrochemical input. So it's not a true cycle. It's not a closed loop. And there's always loss of plastic as well at the product and use stage by humans. So we always have to have an input of plastic. And also some of that might get burnt in waste and other things. So there's always a net flux of oil to CO2 in this cycle. So it's not perfect, but it's very good for PET, not very good at all for many other polymers. So if we wanted to think about something a little bit more sophisticated and using our knowledge of enzymes and synthetic biology, we could then start to think, well, could we use an enzyme to break down the PET and recycle it? Well, people look for that, and only a few species have been discovered, bacteria and fungi, that are actually capable of partially degrading PET into its monomers. And we'll have a look at the monomers, how PET is made and degraded in a couple of slides. The monomers are terephthalic acid, TPA, and ethylene glycol. And we'll have a look at the structures of these in the next slide or two. So here's just some examples of a couple of organisms, Fusarium oxysporum and Fusarium solani. These are fungi, grow in the soil, and people have isolated those growing on what we call minimal media, no added carbon like sugars or anything, but containing PET fibers. They can grow, but they grow very slowly. And then in Japan in 2016, a paper was published with this bacteria, this Idionella sachiensis, and it was isolated in Japan around a factory that made polyethylene terephthalate. And it's able to utilize PET film as its major carbon source. So it actually grows on PET as a food source. but it grows very slowly. There's a picture of it on the left there in A, spreading out from a streak on an agar plate with some PET film there. And then this is an electron micrograph, so zooming in using an electron micrograph to get a much closer picture on B, the panel B there, or the bacteria actually growing on the surface of polyethylene terephthalate. So what the enzyme, what the bacteria is doing, is taking this PET polymer, shown by these linked circles and triangles here, it's secreting enzymes. Now that means it's able to make enzymes and actually secrete them outside of the cell into the environment just around the bacterium, and that chews up the PET polymer. Now the PET polymer is too big to get into the cell, so it needs these enzymes, these secreted enzymes, to chop up the polymer. So you get PET breakdown into monomers, and those are small enough to be taken up by the cell and just used as a food source, as if it was growing on glucose or something like that. Now, if we look at these in a little bit more molecular detail, so this is now understanding PET as a polymer. What is it actually? It's a polyester. So here's the ester group, and you'll probably have learnt in your high school that an ester is a bond between a carboxylic acid and an alcohol. When they come together, water is lost in a condensation reaction, and you get a new bond, this ester bond. It's quite strong. And here in this polymer, it's repeatedly used. Here's one in the circle, but there's another one here and here and here, if you can see my pointer. So it's a repeated use of these ester bonds between two compounds. And the compounds are here at the bottom: terephthalate acid, very symmetrical carboxylic acid, and ethylene glycol. And it's these two materials that are polymerized to make polyethylene terephthalate. And here we've got a little bit more detailed structure in the benzene rings here. We've got the oxygens in red, and we've got this repeated polymer. And you can see how some of the properties of PET are inherent in some of the molecules that make it up. Terephthalate acid, benzene ring with these symmetrical carboxylic acids, is a rigid, flat molecule. And the ethylene glycol provides a little bit of a rotational linkage between these rigid, flat molecules. But if you get a lot of these polymer molecules, these benzene rings will tend to sort of lie on top of each other and stack on top of each other. They interact and they'll form a nice crystalline polymer. And that's why PET bottles are tough, even when they're very thin, and they're crystalline and we can see through them. Very good properties. So if we wanted to break this down, we need an enzyme that can cut this bond here, this ester bond. Now in nature and in human bodies and in plants, there are many esterases. These are enzymes that cleave ester bonds by adding water across the bond. That's a hydrolysis reaction. And many, many types of esterases exist in nature. Some will break down fats to their long chain fatty acids and glycerol. So they're very prevalent in nature. Many other esterases exist. And the enzymes that break down PET are esterases. And one is called, we call PETase, just for ease of saying this long name. And the other one is MHETase, because when PETase acts to break one bond and then another, very often it's not this bond that's cleaved second of all, but another one here, releasing this molecule MHET with one of the ethylene glycols on. We can also get release of the central bit with two ethylene glycols on. So we get a mix here, and obviously terephthalate acid and the ethylene glycol. So the organism has two enzymes, a PETase and an MHETase, to give these two final compounds here. And then these can be easily taken up and the bacteria eats those as a food source, but it's very slow. So what we want to do is to make this faster and better. So the bacteria secretes these hydrolases. And I put this word up here, cutinases. Now, anybody who wants to do any more sort of further interesting digging on this, look up cutinase, and you'll find out that those exist in nature in bacteria that can eat certain kinds of things from plants. So you could look up that and have a little bit of a journey into what cutinases are, because the petases seem, now that we know their enzyme structure, they seem to have evolved from these cutinases that existed for millions of years before PET was introduced into the environment. Now remember, we're thinking about an enzyme and a bacteria that is able to grow on something that is a non-natural polymer and didn't exist in nature 50, 60 years ago. So for the millions of years that bacteria have been around, or billions of years in Earth, they would have been using enzymes like this to do something else. But it just so happens that a cutinase, A and B, has the right kind of esterase characteristics to be quite able to start degrading the PET polymer. And then this organism, this Ideonella sachiensis, has obviously spent quite a few years growing and dividing and growing again on the PET polymer, and it gradually, a cutinase has evolved into a PETase, and it's got an associated MHETase as well, which has evolved from another esterase. So you can see how bacteria have great versatility, and they can actually be evolving and growing on some of these man-made polymers. And this is what's been exploited now to turn this into a process for recycling PET polymers. So that's what we've been doing. We've developed this plastic waste innovation hub at UCL, where we're looking at different plastics and how to degrade them. And we started looking at the degradation of PET using enzymes for recycling back into bottles again. But this molecule here, being an aromatic ring, has quite a lot of other uses. And what we're doing is we're taking other enzymes that we've got in our portfolio, if you like, of enzymes in our group that we've built up over the years. And we're able to then use enzymes stepwise to take these into higher value products. And that could be the fine chemical, intermediate chemical market, or pharmaceutical sector. So that's what we're doing in the Plastic Waste Innovation Hub research. And what we want is new enzymes. So we want to get new petases that are better, and MHETases. And we use genome mining, so we want to look into the DNA sequence, the genomes of new bacteria. So we need to isolate new bacteria. And we're also very interested in thermostable enzymes because they are much more robust for being used in industry. So this is quite a complex slide. This is what we're doing in our research. So Maria Bourne was shown in one of those early slides. She's a postdoctoral researcher doing this work. She's looking at the genes for these petases and mutating them in the lab, and then putting them back into bacteria. We can then look at these as colonies on an agar plate, and using some automation techniques, we put mutant bacteria, each with a different mutant gene, into these little wells on a microtiter plate or a microwell plate. We do some interesting color screening with some chemicals that we've made that are what we call chromogenic, that mimic the PET polymer. And after that, we can find a few evolved or modified or better petases. So this is a complex slide, and if you were studying biochemical engineering, you'd be learning about what all these stages mean, and some of these are things we do in some of the practicals in the lab. So what we've been doing is looking for new bacteria. So we've taken PET bottles, food containers. This was on the floor of my office late last year. We shredded them, cut them up into very small pieces. We put them into this hot bin. composting unit. Now this is a thermally insulated composter. So it allows the bacteria to grow and heat is retained. So it means the microbial metabolism that generates a little bit of heat, that's all retained. So the temperature grows to usually 50 degrees or more. So it's called a hot bin composter. So things can degrade very fast in that. And we've added PET plastic, but also some PET microbes that we've isolated in other systems to this, and we let it grow for a while, and we're isolating bacteria and their DNA. So we then do the sophisticated molecular genetics to sequence the DNA of these bacteria, and from that we can discover new Appetizers and MHETases, the genes and then the enzymes. And this is a little bit of, again, some of the material that we'd use in the first year to teach you how we do molecular biology. How do we clone genes? We use, or often use, these circular DNA molecules, very small ones that can exist in bacteria as multiple copies. They're called plasmids. We also call them vectors to carry new DNA fragments. And this slide, I'm not going to go into all the detail here because this is something taken from one of the first year lectures. So we're using restriction enzymes. This one's called Ecoli. This chops up DNA. The vector has a single site for the enzyme, but the DNA from the source, that could be a PET degrading bacteria, has many thousands, probably millions of sites, chops it up. This is showing just a small fragment of this, into fragments, and each of the ends of these fragments is the same as the end of the vector. We can use a DNA joining enzyme. They base pair these fragments here because there are sticky ends caused by the asymmetric cutting of DNA by this enzyme. So again, these are all terms and techniques that will be discussed and described in much more detail in a first year lecture. And what we make are recombinant plasmids. So this is recombinant DNA technology in a very sort of small nutshell. A vector being opened at a single site, DNA being cleaved by the same enzyme, sticky ends being joined by a ligase, and we make a new circular molecule. The circular molecules can be put into cells, and that's what's being shown here. We take Ecoli, for example, our little lab bacterium that we do a lot of our molecular biology in. We treat that with calcium chloride, and we add plasmid DNA, and that can go into the cell. And the plasmid DNA comes from this experiment where we're joining DNA from a Appetizers bacteria with a vector. That's going into the cell. A transformed bacterial cell will then be put onto an agar surface and allowed to grow overnight with an antibiotic. All the cells, only the cells with the plasmid can survive. And then we've got each of these cells containing a different plasmid with a different fragment of DNA. And that's basically how we start to isolate the gene for a particular property like a Appetizers enzyme, and then we can start to study that. Okay, we're back to something more sophisticated here. We can make these libraries, that we call them Appetizers, and we can use ways of mutating them, automation to pick out the colonies, and we can screen with different activity screens to look for brand new enzymes that are better or different properties. We do the DNA sequence of them. So this is very sophisticated work that only PhD students and postdocs would do, but this is going on in our lab. And so we've got new Appetizers and MHETase enzymes that have better properties than the published ones, and we're gradually starting to use those in larger scale. And by larger scale, I mean we need to grow a lot of the material. And that's what's shown in this slide here. Going back to what we're doing in the previous slides, we've got these plasmids and the vectors and the genes, putting them into the bacteria. And once we've decided which of the bacteria have the right genes, or we mutated them, we do a culture workup. This is usually small volumes, 5 to 10 mils. They go into a small scale shake flask, 50 mils, for example, slightly larger shake flask. And then if we were going to make a lot of an enzyme, we'll put it into a fermenter a bit like this. This was a 450 litre fermenter that was in our Advanced Centre for Biochemical Engineering. And this is what biochemical engineers do. They do the fermentation, that's the growth of these kinds of bacterial cultures, and what we call the scale up. So once you've grown a large amount of bacteria, you can then extract the enzyme and start to use that as a way of degrading PET. But we use this fermentation and scale up idea for studying lots of proteins that we're interested in, or making therapeutic proteins, for example. So here's the last slide, and it's really taking a couple of steps forward into what might be possible in the future. So plastics or polymers are very valuable, and I don't think we want to think of just getting rid of them all and not using them. What we need to do is to think about where are the problems in their use, and I've described some of those. It's their end of life. And if we start to think about the whole cycle of use of materials in the future, from beginning to end, a proper life cycle analysis would say, well, let's think about how to actually use the polymers at the end of life. And if that's going to be done, let's think also, can we make the polymers from some sustainable source? And already many different groups, including ourselves, are looking at biomass. So that's waste agricultural material. So all the stems and stalks of crops, agricultural crops, or things like the waste after sugar extraction from sugar beet or sugar cane. We call that biomass. It's virtually free to use, or it's very cheap, and doesn't have many other uses, but it's captured sunlight and captured CO2. Biomass can then be made into polymers via breaking down the biomass to sugars, for example, or if it's lignin, into the aromatic components, and that's chemical and biochemical engineering. And then we can build biodegradable polymers. So there's a lot of interest in this whole area, and we're doing this, trying to build up new biodegradable polymers. And what we want to do is to match the kind of properties that the current oil or petrochemical based plastics have, and we're able to do that. And there are some good examples of biodegradable polymers that have as good and sometimes better properties than existing oil based ones. Now these would then be compostable because they are biodegradable, so that means you can put them into a certain kind of compost and degrade them. We can make them into packaging first, but when it's end of life, we can collect them, we can sort them, compost them, and these degraded parts can either go back to be built into polymers, or they can be used just as fertilizer, biomass, and grow again. And we have a proper cycle. But you note that the input here is CO2 from the atmosphere, sunlight. And so any kind of escape from this cycle into the soil is captured, because the soil will then secrete CO2, and then that goes around the cycle. And this would be a possible scenario for quite near in the future. This is already being developed in many parts of this cycle. Okay, well, I will stop there. I've taken a little bit longer than the 30 minutes. Thank you for your attention, and any questions. I think I will go out of the screen share, and I think then we go into a questions session.
Sure. I'm just going to do a screen share, so bear with me a moment. I'm going to try and find my hang on. Yeah. So I'm going to put up a page with some contact details in a moment. In the meantime, we've got 9 questions open in the Q&A section, and a couple of ones in the chat box as well. So bear with us a moment. John, are you able to see the Q&A box?
I just opened that, and I've got some questions here that I can see. I don't know how they, oh yeah, they've got the times next to them, so I can try and answer the first ones first. Would it be possible to synthesize an enzyme dedicated to the polymer substrate? Yes. So can you all hear me? I won't type the answer. Do I have to do that?
Yes, absolutely, thanks.
I'm doing this. I'm not used to this particular connection. Yes, of course it is. Now, the question actually embodies quite a few levels of sophisticated molecular biology. What I described in the work that we're doing, and others, we're trying to find new enzymes that are out there in nature, either by enrichment from microbes that might have those enzymes or might develop or evolve them in a hot bin or composting like that. Now that is one way to find an enzyme dedicated to a polymer substrate, because you're actually enriching it, the microbes that eat the polymer, on that polymer substrate. And if you go through a few months of that, then there's quite a lot of evolution going on in that hot bin composting. To synthesize an enzyme from scratch in the lab, we would, now that is possible, it's quite a lot more complex because you usually need a good structure, an idea of an enzyme that does work to build that on. And what we normally do is take an enzyme that might have the right cleavage properties. So I'm going to use an example of nylon. Nylon has amide linkages. Now, there's a lot of amidases and proteases in nature. All our food that we eat, if it contains proteins, we have to degrade with proteases, and they're degrading amide bonds. So it'd be quite possible to take a known protease and develop that into a nylon degrading enzyme, because the bond it's cleaving is the same. Now, obviously, the material around that bond in nylon is different from the amino acids in a peptide chain, but it's not very difficult in the lab over a course of a year or two to develop that kind of enzyme. So the answer is yes, it is, but there are various routes to that, some a little bit more complex than others. Well, I hope I answered that to the depth that you wanted. Okay, the next one, how can a bacteria consume TPA, since TPA has relatively low solubility in aqueous medium bacterial cells? Well, that's very true, that's very perceptive, but bacteria living in the soil will also consume a lot of other low soluble material. For example, the breakdown products of lignin are rather insoluble. But in the soil, you have to then imagine there is intimate contact between bacteria and their substrates, usually. And what you find in nature is that if you put a branch into the soil, it becomes coated with bacteria. So they're in intimate contact. You don't have to think of, oh, you have to have soluble things that migrate into solution and then diffuse out for long distances. There's a lot more intimate contact. So although the solubility is low, bacteria can cope with that because they are virtually living on top of the substrate and sometimes able to sort of degrade and take it up across a few nanometers. So that's an answer to that. But also, I mean, bacteria can grow on very low concentrations of material in the soil or in solution, and they grow slowly. That's really all that happens. Okay, would it be possible to use synthetic? Am I live on that? Do I have to click the box answer live on each one, Kim?
It's fine, I can process these, but then just to let you know, there are 12 questions, so it might be worth having to scroll through them, just in case there are some that are relating to the same subject. You might be able to answer them together. Also, there are a couple of questions that have come through in the chat. Somebody's asked, for example, they asked whether you could create a polyester that dissolves in water but doesn't affect the ecosystem or have the water's condition, or affect the water's condition. Somebody else has asked why we don't genetically manufacture specific enzymes, and whether they've actually already been, whether anything has happened on an industrial scale like this at all.
Yes, well, industrial scale, yes, that is something. Over the last year, a company called Carbios in France has developed a very robust PETase process, and it's now teamed up with some large groups like Nestle, Coca-Cola, and a couple of others, L'Oreal, and it's now developing a really large pilot plant to consume PET with enzymes and recycle the polymers. I might just quickly go through some of these things. Would it be possible to use synthetic catalysts to break polymer chains? Yes, but bacteria make the enzymes just by growing the bacteria. And synthetic catalysts have to be made by organic chemistry, and eventually that is more expensive than making an enzyme and more malleable, and we can make enzymes of different kinds to cope with different polymers. Can this method be applied to other polymers aside from PET? And that's what I described with the nylon. Now there's a lot of research going on for other polymers, polyurethanes, like lycra, nylon. So yes, the answer to that is there are now people trying to do this for a whole variety of other polymers. Polystyrene, I've noticed recently in a publication with some degradation of that. The answer to Gabriella, how effective is using synthetic biology to recycle plastics on a large scale? So that example of Carbios I mentioned is now growing to, I think, a $10 million plant that was built. And I think you'll see now in the coming year that being talked about on a very large scale and extending to other countries, and also then extending to other plastics. So as the years go on, and in certainly this coming year, you'll see that really quite large growth in that area. Why focus on PET and not another plastic that's not easily recycled mechanically? Well, the reason why people focused on PET is that it's easy to capture and the waste, the domestic waste. We already put that into separate recycling containers, and then in the factory that can be recycled and picked out of the waste much more easily than others. So some of the other materials or it's the companies that do the waste recycling, are starting to work on how to separate those from the waste, and then it's up to the sort of polymer recycling enzymology to work on that. Why can't the recombinant plasmid? Well, we do that. Usually in a situation where you've only got PET to degrade, you only want the PETase, but that is also being used to secrete the metase as well. So the answer is that yes, that is being considered. So you might then end up with organisms that could actually eat several different polymers. All of these need to be trialed and tested, but all these ideas are very legitimate and have been addressed or being addressed already. If the cost of production with plastics and glass is the same, well, glass uses a lot of energy. Every time you melt it, has to be heated to, I don't know, 800, 1,000 degrees. It's hugely energy intensive. I think plastics in their total energy use is less, but it's very difficult to get a full energy balance if you go all the way out to the recycling at the end and the cost of getting the oil shipped. But I think what we need to do is start to consider the full cycle of recycling back to the beginning again with very little losses. But the inputs, if we can have them as biological, then those are the best. Why not use a virus for transfection? Well, that's just a different methodology. Viruses as vectors, plasmids as vectors, it depends on the research group using those. Yes, there are different methodologies as that. Well, we use fed batch for the cultures. we have research going on in the department on chemostats for a different process that's actually more complex. And you'll learn something about that if you join a biochemical engineering department, because the difference between those two growth systems is quite complex, and chemostats are actually more difficult to run. But we're looking at that because ultimately that is cheaper. With this, I suppose, Jessica, you're referring to maybe that final slide. Well, ultimately, I hope it could be close to 100%. I don't think anything can ever be 100%. You get thermal losses. Of course, it's difficult to have anything being 100%. But that is an idea to try and make it as close to that as possible, and using inputs from nature as well. What are the other valuables? Well, phthalic acid, well, the aromatic part of a molecule can be built into many other things. And most drugs that we use, new pharmaceuticals, will have at some point some aromatic part in their, or moiety in their structure. So yes, we would have roots from that to make other chemicals, particularly the amines that are derived when we start to put amines onto those. And we can recycle via transesterification, yes, but there are many other valuable uses for the aromatic part of that. Current limitations of using biomass, well, it's often the processing of the biomass to begin with. Often it's dispersed across many different agricultural sites, so you need to gather it. So there's problems there in getting it all in from a certain point, and then the degrading of that using enzymes. But there's huge amounts of work going on that around the world, so that's the limitations are being addressed by many other groups. Can we utilize commercial process making pet degradable value added products? I think I've answered that with a variety of the answers to the other questions. So if altered bacteria ever released into landfills to degrade plastics, what could possibly happen? Well, I don't think they would harm the environment, because they're actually, we now design enzymes and microbes for a process, and that process is often quite specific for a factory. So the enzymes we're making and developing are for high temperature use, 50, 60 or more degrees, and often in very high concentrations of the substrate. So the enzymes just wouldn't work in a normal landfill, simple as that. What of the examples of biomass have been used so far to produce biodegradable polymers? So anything that can be made from cellulose into glucose, and that's many different types of biomass. The glucose can be used in a process to make furans and other kinds of derivatives like that, which can then be polymerized. So anything that can be made into glucose can be converted to other biodegradable and bioplastics. We can also use lactic acid grown by lactic acid bacteria on glucose, and the lactic acid can be polymerized into polylactate, and that's a biodegradable, high-value biopolymer. So quite a lot, and we're looking into even more types of biomass that can be degraded, like sugar beet is another one that we're starting to look at. But around the world, much of the work is going on how can you convert glucose into lots of different biopolymers. How can this be applied in cases of pre-existing pollution, for example, plastics in the ocean? Well, it is difficult because you can't just put organisms into the ocean. First of all, that's not allowed, and also they wouldn't survive and they wouldn't catch the plastic. In some ways, we have to think of how to collect that. And there are ships going out there to try and corral plastic in these giant ocean gyres that exist in a few places and maybe collect those. Now that is a massive problem for perhaps you and your cohort to solve in the future. Our iGEM team, the student-led iGEM team here that do summer projects in the iGEM worldwide scheme, have looked at this several years ago. But those are exciting things to think about for the future. Still quite a big problem to solve. Well, Shri, I think I've answered that. You also, can we have enzymes that hydrolyze every available? Yes, that would be feasible and possible. We might want to do it one at a time so that we extract each of the nice monomers for use instead of making a great soup where we have problems in trying to get things out. I've answered the thing about microplastics. We're not allowed, there are rules against releasing engineered bacteria into the environment. What would be necessary is to gather that material together, but that's something perhaps for solutions in the future. I think that's the same as the answer for Nick. It's in esters, we have to gather it together. What are the first steps? Oh, where's that gone? What are the first steps that we could work on, develop a research? Well, we just need some industrial money and startups to get together. Maybe that's something for younger people to think of. I'm getting too old to think of starting as an entrepreneur, but I know actually there are several PhD students and postdocs now starting to think about this, either to try and do what Carbios are doing or to try and go for some of these other polymers, plastics that we use. And my work and some of the work in the group is looking at things like polyurethanes, lycra, nylon. Could other organisms be used to break down polymers, for example, fungi? Well, of course, yes. It really depends what's the best at doing that particular job. Has a fungi got really good enzymes that the bacteria doesn't? Then we just simply start to investigate and start to use the enzymes from the fungi. Quite often we take the genes from these organisms and put them into a bacteria that can grow faster. But we could also put it into a yeast or a fungi. So I think I've gone down that list of questions. Now, Kim, were there questions from anywhere else, or have we finished our allotted time?
I think, well, we probably would have time for one more question, but I think we've gone through. If there's any question that we haven't answered, by all means, post it again. It's been an extremely well-attended seminar, so we've had loads of questions, which has been great. But if we have missed anyone out, please do let us know. Otherwise, it's probably worth mentioning, John, perhaps you want to say, if people wanted to pursue this further, there's the MRes, and what modules in which degrees would people be able to find out more about this subject?
Well, this particular subject is what the MRes in synthetic biology, but also the MSc in biochemical engineering would have parts of this kind of problem and descriptions of how these might be solved within it in some of the projects, for example. And what I did in the some parts of the lecture, I talked about the examples that I was using are slides that I took from my first year lectures. So when we talk about how to clone genes, how to express them, that's all covered in our first and second year biochemistry and molecular biology for engineers lectures. And you also learn about how to look at scale up. What does scale up mean? What are the different ways to do it? Answering that attendee's question on batch fermentation versus continuous fermentation, that's addressed in first and second year lectures. So a lot of the things that go to make a whole process are then looked at in detail in the biochemical engineering undergraduate course, the BSc. And that would be the starting point for people wanting to look at these in any kind of detail, because you have to learn all the ways of addressing the problem. The life cycle analysis, the molecular genetics, the enzymology, the fermentation, the downstream processing, all of those go together to make the full process. And it's only after learning all those can you then start to be really inventive as a biochemical engineer to then invent a new process. So I hope that answers the question.
Okay, that's great. Oh, somebody's just asked about whether you can do a BSc and go into biochemical engineering. The very quick answer is yes,
Yeah,
there are ways of being able to do that.
absolutely.
If you want to know more about any of our programs or admissions, we do a series of events for questions about specific programs, and we can have a proper conversation with you about it then. But we have come to the end of our allocated time. Some people have said thank you to John. John, I want to pass their thanks on to you. I've really enjoyed this, and the feedback we've had is great. So what I'm going to do is I'm going to stop screen sharing, and so bear with me a moment. So I'm going to stop screen sharing, and I'm going to send a very quick survey round to people just to ask for a little bit of feedback on this event. But essentially, and that'll come around in a moment. So,
okay, I'll just say thank you for everyone for attending, and the great questions. They're really quite pertinent and really on the ball, and actually are things that we're currently asking ourselves and doing research on. So great questions, and thanks very much for your attention.
Thanks very much indeed. I'm just going to send this to, there's a quick survey here. If you get a moment afterwards, there are only about 4 questions and it would really appreciate your feedback. You do also have my email address. If you've got any questions about this event, anything you'd like to follow up on, please do get in touch and we'll be happy to answer your questions. But thank you very much again for attending, and yeah, hope to see some of you in the future. Cheers.
Okay, bye-bye.
Bye.